All right. Well, thank you so much for the opportunity to present today. I think the first thing I will do when going back is develop a patch, because I think that's awesome. So I'll be talking about nutrition, genes, longevity, and chronic disease. My faculty disclosure is I do some preclinical consulting for Nutra, essentially developing fasting mimicking diets. Is this all cropped for you? Yeah. Sorry. The learning objectives essentially focus on the role of nutrition and nutritional genomics on longevity. I'm going to discuss some of the key nutritional cascades that relate to longevity and health span, and a little bit on genetic variation, although my main focus will still be the role of nutrition, chronic disease, and longevity. We'll establish some interventions to improve longevity. The second half of my talk will be focused on the fasting mimicking diet and its impact on health. So essentially develop an idea to sort of provide a nutrient-based interaction to make healthy aging a more clinical relevant focus. So first things first, I think it's important to really make sure that I'm not talking about these four topics separately. It wouldn't make any sense. Ultimately, nutrition, the genes, chronic disease, longevity all work together. And that's the beauty where my work is. Biogerontology utilizes all these factors and essentially establishes relationship within these four components. And I've been doing this for a while. I started working on yeast maybe 10 years ago, and then I joined the lab at the University of Southern California under Walter Longo, who was, I guess, supposed to be here, but he's unfortunately really busy. So I get to talk to you, which makes me happy. Biogerontology utilizes all this, and so I get to play around with all this a lot. And it's awesome because it's not just one thing. I'm not just talking about nutrition. I'm not just talking about genes or how to live longer, healthier, but all of this in combination really makes an impact. I'd like to start off with this because we can actually learn a lot of this from this cartoon here. On the very basic level, you can see two different curves describing different experiences and how we age. The bottom one, this dark blue line, is essentially this unhealthy lifespan, right? We have this trajectory early on in our 20s already, where the decisions that we make with regards to our life, meaning being stressed, fatigued, having poor and unhealthy diets, as well as being sort of like a couch potato, sedentary lifestyle choices, put us on a trajectory where we increase the risk of developing aging-related diseases earlier. So we essentially get to a point where we live our life with multiple diseases. So the years lived with disease increase by having these poor choices. On the other hand, we can be on a sort of like optimal health span curve where we're active, we eat healthy diets, we progress in being independent and active well into an old age. And then at one point, sort of like our lifespan compresses and we drop. So the idea is sort of like this square curve. You live happy with a high quality of life into an old age, and then you drop and it's over. So again, in this top curve, the years of living with the disease are reduced. So it's essentially, I'm not proposing longevity as a mean to simply live longer by all means. It's useless if we can't improve the quality of life with it, so health span. Another thing that you can see here as well is sort of the concept of chronological aging versus biological aging. If you want to, for example, focus in the middle here, these people in the 40s, right? They're the same age. Chronologically, they're the same. They have the same age. However, with regards to risk markers that predict aging, biological aging, the person at the bottom is way worse off. So the chance of developing aging-related diseases increases compared to the person on top. I also want to stress out that even though I will be talking a lot about nutrition, it's not ignoring the fact that there's multiple other factors that predict how we age and the quality of aging with it. So at the bottom in this colored spiral, you essentially see exercise, rest and relaxation, social interaction, intellect, all this also plays a role in how we age. But the focus really for today will be on diet and nutrition. So, brief cartoon, I guess. You see this person here, bad habits, right? It's all the poor food choices we make, high sugar, sweetened beverages, desserts, junk food, resting at home, all this increases the prevalence of obesity and overweight in the United States and throughout the rest of the westernized world. Then leading essentially to many of these chronic diseases that we see, right? The main top 2 killers in the United States are linked to obesity. It's cardiovascular disease and cancer. So these lifestyle choices predict chronic disease and the quality of aging. We see the same bad habits in a cohort of subjects with growth hormone receptor deficiency that we followed in Ecuador. You see one of them here eating an entire cake. And that was when they actually visited us in Los Angeles. So you see Walter here together with Jaime Guevara, who's a collaborator down in Ecuador. And we had about 25 subjects coming up, and we did a bunch of measurements on them, including MRIs and DEXAs and all that. And you can see they actually, they're obviously small in stature. They have a deficiency in the growth hormone receptor, and I'll come back to that in a second. But you can also see when you look at their body type, they're all fairly overweight and obese. But the really interesting thing is that in their case, this is not related to cancer or diabetes, right? When you compare these subjects on the right here, the growth hormone receptor deficients, to their relatives, these subjects do not die of cancer. They do not get diabetes, which is absolutely remarkable when you look at what their actual physique is like. They should be dying of this, but they do not. And it's compared to their relatives. I mean, you see 20% of cancer incidences in their relatives. They're not genetically protected compared to their relatives, but the deficiency of your growth hormone receptor is really what protects them. The mutation that we commonly find in there, so again, genes playing a role, at least with regards to this Laurent syndrome, it's usually a splice site mutation, which results in a shortened protein. You see the great majority have this E180 genotype, leading to low levels of IGF-1, but also having a better or improved insulin levels and an improvement with regards to their HOMA. So glucose metabolism is improved, again relating to this low incidence of diabetes that we see. So I want to go back and comment on why growth hormone receptor. Why do I start off with this? Well, it links essentially into these evolutionary conserved nutrient-sensing signaling cascades that we see. So when we go from yeast through worms and flies, mammals, all these model organisms of aging, we essentially see that the same pathway exists depending on what you look at, the different names, but they also ultimately all have the same function, which is when nutrients are abundant, we activate a cellular signaling cascade that promotes growth. So glucose is around amino acids. In mammals, obviously this is regulated more through hormones, but all of this results in cellular growth, cellular proliferation. But why is cell growth associated with aging? Well, you could go back into the 1960s and think about Hayflick saying essentially a cell has about 40 to 60 divisions before it becomes senescent and dies off. So the activation of these pathways sort of promotes senescence. On the other hand, we have also shown over the last 100 years that modulating these pathways with, for example, caloric restriction, dietary modification, so you can reduce the protein intake, you can induce sort of like a fasting light state, these pathways become deactivated, which in turn promotes cellular resilience. So the enzymes playing a role with regard to detoxification, heat shock proteins, all this becomes upregulated, essentially providing an anti-aging mechanism. So really, these pathways, you can imagine them being a switch. In the on state, everything growth, it's pro-growth, pro-senescence, pro-aging. On the other hand, turning this off promotes cellular resilience and stops aging. Examples of modifications of these pathways, you can see in here. It's, again, similar to the subjects with Laurent syndrome that I showed. On the right side, you can always see the smaller organism being a mutation within the signaling cascade. So yeast, flies, and mice. The mice that I'm showing here are Ames dwarf mice. They actually hold the record for longevity extension, 40% longer. The notable thing, again, is they do not just live longer, they are healthier. So these mice essentially do not develop any of the aging-related diseases that you would see, the ones you see at a significant lower rate. So these animals live longer and healthier and better. With regards to humans, I think a prime example for the role of genes in longevity and chronic disease are centenarians, typically defined as a person living 2 or beyond 100 years. And obviously, because it is such an exceptional trait, one in about 8,000 people lives to about 100 years. It plays a huge role with regards to identifying genes that are involved in human longevity. There's 2 examples here. The oldest validated case, Jean Calment, she died at 122 years. And as you can see in the picture, she was not particularly a friend of living healthy. So she smoked well into the over 100, and anecdotally, she only stopped smoking because she could no longer light her own cigarettes. So she did not want to be dependent on other people, so she quit smoking. The other one is actually a lady from Japan. And again, you can see here she's still doing interviews at 115 years old. So she's mentally quite fit. There's some physical limitations, but she's there. It's again indicating that just because you live to an old age doesn't mean that you have to suffer from the aging-related diseases. There's some really cool data coming out of these populations when you look at centenarians. In this case here, they even went a step further, and within the centenarians split them up, 100 to 104-year-olds, so these guys here, 105 to 109, and then what they call super centenarians, so it's over 110 years old. And what you see here is essentially survivors, meaning people that had a disease but survived with it, compared to delayers, meaning they essentially got the disease but he got it later, versus escapers. And you can see this shift, right? The people over 110 years essentially never got an aging-related disease. Like 70% of them never got one. Whereas the more younger centenarians, I mean, they're still very old, a lot of them are just delayed. So when you look at some of the data from near Basilai's lab at Albert Einstein, the great majority of these centenarians, particularly in this population of Ashkenazi Jews that he's following, they essentially get aging-related diseases, but they get them about 30 years later. So you would still get cancer, but 30 years later. This really relates to, at the bottom here, the colored graph on the bottom left, when you compare the controls, people they're considered non-centenarians, but they live into an older age, so about mid-80s or so, and then compare centenarians, semi-super-centenarians, and super-centenarians, the percentage of life lived with a disease is dramatically reduced. And this is true for all the aging-related diseases that were investigated. So you can see the age of onset of cancer and here, a cardiovascular disease, dementia, Huntington's, osteoarthritis, and stroke. So even within that population, of a very healthy population, you still see differences based on the cohort that they were grouped in. All of them, compared to the controls, obviously have dramatically reduced risk of developing any disease. This is Walter when he went to Italy, and he visited Salvatore Caruso, he lived to about 110 years old, and he and Walter actually sang some songs together. It's recorded. It's kind of fun. But Walter asked him, like, what do you think you got to live to 110? Or at this point, I think he was like 108 or so. And Salvatore said, no wine, no tobacco, no Venus. So he essentially excluded all the fun stuff. But then when people ask centenarians, why do you think you live to an older age? This is what they say. So a great majority says good genes. Centenarians traditionally come from a family longevity. People live longer. So the relatives of centenarians live longer than non-centenarians. So you see about a third or so say that they come from a good family. The second one right there is diet. So it's already indicating they understand, they feel like they're eating better, they come from an area where their food consumption is associated with health, but it's actually not necessarily true. So, so you see these, you see a 100-year-old smoking a cigarette, which is fun, but the red boxes indicated one thing to begin with. Exceptional longevity is a trait often found in women. There's more old women than men. But when you compare people with exceptional longevity to a nationally representative data set, there are no significant differences other than the cigarettes smoked in men with regards to some of the risk factors that we would consider now, or protective factors that we would consider nowadays to be pro-aging, right? So you see in the number of cigarettes smoked are comparable, alcohol consumption is comparable, physical activity, low-salt diets, low-fat diets, low-calorie diets. There isn't really a dramatic difference there. So it is, for the case of centenarians, their perception of it is that they live healthier, but they don't. And it's actually, these people are 100 years old. Some of them have seen 2 world wars, and they were certainly at a stage where none of the physicians recommended, like a low-carb diet to them. They ate whatever they wanted. And there's instances of like hard labor, or even in some cases they were obesity-related in their 30s or 40s. But none of that matters to them because their genetic code essentially puts them, even in a crappy environment, they're protected. So they can really, you can put them somewhere, and within that environment they do well. So the idea really is that they're not distinct, at least centenarians, not distinct in terms of lifestyle factors, but it is more their interaction with environmental factors. And when you look into this cohort of centenarians, it goes back again to some of these components of this axis that I showed you before, right? You see with older age an increase, for example, in FOXO3A, that's the red line right on top, essentially a transcription factor downstream of the signaling cascade that I started to talk with. And a lot of the traits that are associated with longevity are all within nutrient and metabolic pathways. So again, you see insulin sensitivity being up there, antioxidants, the IGF-1 axis multiple times, fat lipids, and so on. So it is when considering the cascade data showed and the data here, I think it's really clear that nutrients, the response to nutrients within the cell modulates the, at least in centenarians, their benefits, and which relates them to longevity. In comparison to those, we still have hotspots around the world where people live longer. They're just not considered a centenarian, but these are communities that outlive the rest of the world. These are the current ones, so starting obviously with Japan and Okinawa, to going through the Mediterranean areas, Icaria and Sardinia. Then there's a population in Costa Rica, and then interestingly for us, there's another one in Loma Linda in California, which is the population of Seventh-day Adventists there. So there's multiple factors for the Adventists. It's not just diet, they obviously have a lot of other factors, but from these blue zones, again, we can learn how to improve the aging experience. And some of the factors are shown here. So you want to maintain physical activity. There's sort of like the purpose in life, a downshift, which is obviously stress, fatigue is reduced. There's a little bit of an indication that they consume less calories because they tend to consume a bit more nutrient-dense food. Plant-based diets, wine at 5, which I think is a great idea. And then you sort of have this community aspect of it, right? So you have this like right tribe, being born in a right family, in a right environment, and religious beliefs to a degree. And the interesting thing is, even for these Adventists who outlive even the Californians, which some of them are real health nuts, so the the adventists live longer, and when you compare within this Adventist cohort subjects that actually adhere to a vegetarian diet, a semi-vegetarian / pescatarian diet, and non-vegetarians, you look at risk factors associated with metabolic syndrome, even in these healthy populations, you can now start to filter out dietary patterns that are associated with improvements in their health. And you can clearly see that at least the vegetarians and the pescatarian diets do a lot better than the non-vegetarians with regards to metabolic syndrome risk factors. So particularly, obviously, HDL, triglycerides, glucose, blood pressure, BMI, waist circumference. And this, again, relates to life expectancy. And you can see in the red box here is essentially the Adventists. At the bottom ones are vegetarians within this cohort. And right above the red box is, for comparison, the life expectancy in the United States. This is a little older study, but it's still true. And then it's split up sort of like on top to bottom. It's essentially men, women, and then life expectancy at birth versus the life expectancy at age 65. And you can really see that there's about a 5-year improvement for the man and an even bigger increase within the vegetarians compared to the US cohort. So again, lifestyle factor matters, diet on top of all this matters. So links to diet. I think the main diet that we all would, off the top of our head, identify with what is a healthy diet would probably be the Mediterranean diet. So defined by usually high intake of fruits, vegetables, whole grain cereals, and then obsessive amounts of olive oil. My boss actually recommends 80 grams a day, which I think is too much, but anyways, and then obviously sort of like moderate amounts of poultry, white meat, traditionally a lot of seafood, fish, then you have legumes, eggs, low-fat dairy, nuts, seeds, and olives, and then really barely any intake of sweets, potatoes, processed red meats. And I think we're all well aware that it is the total opposite of what we consume in the Western diets. So absurdly high amounts of desserts, sweets, processed meat, red meat, processed foods, and then like tons of fries and all that high-fat dairy. And then opposite to what you should really be eating, low amounts of vegetables, legumes, and fruit. So we know all this, the translation into, knowing it doesn't necessarily mean that everybody follows it. And I'm assuming it's also even harder to really implement when you talk to people where social economic status matters or convenience-based foods. There's a reason why you can go to Taco Bell and buy a $5 box and you come out full. You feel like shit, sorry me. 2 days, like 2 hours later, but you could, but it's cheap and it's available and it's on every corner and none of us has time to go and cook anymore. So it's really, that's the trade-off, I guess, that's in there, but we know we shouldn't be doing it. And so it is essentially, we know it and we know that the composition of the diet matters. So by following these diets, you obviously shift the macronutrients around, right? So the Western diets is usually like high protein diets versus the Mediterranean diet is usually like a plant-based, low amount of fish. So protein is reduced, and we see these patterns repeatedly in animal studies where we know that what we feed predicts lifespan. So this is a really nice study done by a colleague of mine at the University of Sydney, Samantha, and what she did is essentially put mice on the lifespan or throughout the lifespan on different diets by simply modifying the ratio protein to fat to carbs. And what she immediately found was essentially that by reducing the protein intake and increasing carbohydrate intake, you improve survival. An example is here. There's like the red peak in this heat map actually indicates the biggest benefits, and you see it's low in protein, high in carbohydrates, and the fat is somewhere in the middle. So we know that protein activates this growth hormone IGF-1 cascade, and therefore is a pro-growth signal. And it has dramatic effects. It's not just a couple days or a month. It's like you can see shifting the protein ratio at the bottom here, you have a 30% change going from the the lower end to the higher end, whereas this area here is obviously a diet that's low in protein and high in carbs. So it's again, we have all this information. Protein matters, and we also know that the source of protein matters, and I will come back to that in human studies right now. So what we did in using a national representative data set, in this case here, was simply compare the level of protein intake with regards to mortality risk. And fairly basic, so the highest protein intake group is 20 or more % of the caloric intake coming from protein. The moderate is 10 to 19%, and then the low protein group, which is sort of like the reference group, is less than 10% of calories from proteins. And I'm fully aware that you can't read this, so we summarize this. And looked in the older population. So in this case, age 50 to 65. Those in the highest protein intake group had a 74% increase in the risk for all-cause mortality. They're 4 times as likely to die of cancer when you compare them to the low protein intake group. And you can eliminate these associations when you substitute plant-based protein for animal-based protein. The interesting thing is when you look into an older population, over 66 in this case, it's actually the opposite. You want to have a higher protein intake diet. And I mean, protein is related to muscle mass. You want to protect older subjects from becoming frail. The moment they're frail, they fall, they go to a hospital. We all know the trajectory they're on. It will essentially lead them to their grave. They become dependent, and it's just... Anyways, so we know at older ages how we absorb nutrients matters. And what we show here is that a dietary recommendation should not just be generic, right? I think what we need to take away from this slide is to emphasize that nutritional needs change with age. It's not just a one-size-fits-all approach. But at least until we get to like an older age in the 50s to 65, the general recommendation for healthy aging should be a low-protein diet. It is further supported. We did another study, including about 130,000 participants from the Nurses Health Study and the Health Professional Study. And again, we see that high animal protein intake is positively associated with cardiovascular mortality. On the other hand, high plant-based protein intake is inversely associated with all-cause and cardiovascular mortality. So again, even though you're still consuming protein, it matters where it's coming from. And it's further demonstrated when you actually substitute either when you substitute animal-based protein for plant-based. You can see this shift here in all-cause cardiovascular disease and others, essentially favoring the substitution for plant-based protein, particularly instead of consuming the processed red meats. So processed red meats are by far the worst thing you could probably eat. So coming back to this, and in the last 30 minutes or so, I will talk about how we can actually utilize nutrition to improve longevity and treat chronic diseases. The idea is sort of say, how can we get back from the unhealthy lifespan back to a normal lifespan, or to an optimal lifespan? There's multiple approaches to this. Again, caloric restriction has been done somewhere in the 1900s, early on, and the effects on developing cancer are well established. Just in the last 10, 15 years, a lot of other approaches have come out. Intermittent fasting, we utilized a lot of water fasting. Now we're doing the fasting mimicking diets. Again, you can modulate your macronutrient balance, focus on proteins. And all of this really provides a nutrition-based treatment approach that you can utilize and that can be implemented with almost any therapy. Part of the studies that I did as a graduate student were based on implementing a fasting mimicking diet throughout a lifespan in mice. In this case, the fasting mimicking diet was a 4-day approach every other week. It's a diet low in carbs, low in protein, relatively high in healthy fats, so like olive oil and coconut is in there, and then moderate amount of complex carbohydrates. I'm not going to go into much detail on the study, but essentially just want to focus on the chronic diseases here. So we used middle-aged mice. They were 16 months old. And every other week, put them for 4 days on a diet, and then they could eat whatever they want afterwards. There was no caloric restriction. There were no control fat. They could eat whatever they wanted. And the first thing that we saw is actually throughout these 2 weeks, they eat about the same amount of calories. So they, after refeeding them, they overeat, and over this 2 period, there's no caloric restriction involved. So none of these effects are related to reduced caloric intake. The first thing that we saw was multi-system regeneration, which was really interesting to us. Essentially, during this fasted mimicking diet, particularly the liver responds by shrinking. The liver shrinks by about 35%. And this is not just water. This is essentially like the cells die off. But then when you refeed them, within 2 or 3 days, the liver goes completely back to their normal weight. And when you stain for markers associated with cell reproduction, you see a dramatic spike. So there's a regeneration that is following this low-calorie diet during the refeeding period. Despite the fact that they ate the same amounts, the adiposity, visceral adiposity, was reduced. What I'm showing here, because I focus on chronic disease, is the incidence of tumors. You can see in red are animals that were maintained on the fasted mimicking diet. We have about a 40% incidence compared to 70-75% that you would see in the control group. So a dramatic reduction in tumor incidence. And on top of that, you can actually see this little bar shifted. The red dots versus the gray dots indicates when we noticed that there were tumor in these mice. And you can see a shift. The fasted mimicking diet is sort of like delayed the occurrence of these 40% of tumors. And considering the animals living to about 35 months or so at a max, this 4 to 5 month shift is fairly dramatic. We see a reduction in inflammatory diseases. We see an improvement we see an increase in the immune and cognitive rejuvenation. We can actually test this by behavioral tasks in animals. And when we performed autopsies and stained the brain, we actually see an increase in neuronal stem cells. And all this together increased median lifespan by about 11%. There was no increase in the maximum lifespan, which at that point was a little unclear to us. But I think going back to the data that I showed for the older humans, the trade-off for this low-protein diet at an older age needs to be considered. And I think there's, in order to improve this more, we would have to increase the protein content again at an older age in these animals. We did some really cool studies in diabetes, essentially reversing diabetes using this fasted mimicking diet. In this case here, I'm showing type 1 diabetes. We have done studies in a mouse model for type 2 diabetes as well. The idea here would be that you treat animals with this drug, STZ, and what it will do is it depletes insulin-producing beta cells, leading to diabetes in these mice. So you can see essentially the blood glucose doubling to about 400. Once that was achieved, we started the fasted mimicking diet, which is indicated by these dotted lines going down. So we did 5 cycles, and you can see that over time, throughout these 5 cycles now, the blood glucose actually starts to normalize, going back to a normal range again. And so when we then stained for insulin production in these beta cells, you can see at the bottom, on the very left, this is what the normal beta cells should look like. The treatment with the STZ essentially depleted insulin-producing cells. On the other hand, after 50 days, the combination of the STZ together with the 5 cycles of the fasted mimicking diet completely restored insulin production in these cells. And we actually did a couple of gene expression analyses on this, indicating, and all of this indicated that we almost have like an environmental induction of gene expression that is associated with embryonic an embryonic-like phenotype. So we kind of like reestablish that these cells actually can produce beta cells. There's multiple other applications for this. We used the fast mimicking diet in the treatment of autoimmune disease. There's an example that I'm showing here on a model for multiple sclerosis. So you can immunize mice, then essentially become multiple sclerosis-like, leading to the onset of a you would model essentially, you can track the progression of the disease by applying this EAE score. So essentially going from 0 is no symptoms, 1 the tail goes limp, 2 would be a balance issue, so the animals wobble a little bit, 3 is paralysis of one leg, 4 both legs, and 5 the animals would essentially have to be neutralized. So, but then you can see the control animals that were just maintained on a regular diet and then treated with EAE all progressed fairly rapidly. So within 10 to 12 days after immunization, they have a fairly severe phenotype. They go from about 1 or 2 hind legs being paralyzed. And we compared this to a ketogenic diet, which is the red one. Ketogenic diet is used because it's sort of anti-inflammatory. And it has improvements, but it's not very dramatic. And then we compared to the 2 different models of the fast mimicking diet. The S indicates a semi-therapeutic approach, where we essentially started the diet once 10% of the animals showed a symptom, which is the tail going limp. And the T indicates therapeutic, so the animals started being treated once each mouse had a symptom. So the moment the tail goes limp, you put them on a diet. And you can really see that there's a nice separation where treatment, 3 cycles with the fasting mimicking diet essentially significantly reduces the progression of this multiple sclerosis-like symptoms. And they maintain this low level. So after the 3 cycles, they don't go back. We also tested whether it's just an effect of them being caloric restricted, and it's not. Caloric restriction essentially looks a little bit better than the control, but not by much. We also used the fast mimicking diet in this model to test whether it is an actual treatment. So we used the control group. We essentially let them grow out up to this point here when they were at their highest peak, and then started the diet. And you can see the animals essentially going back down again. So again, preventing a treatment modality. The whole idea of why we initially developed the fast mimicking diet is actually based on cancer treatment. So a lot of the studies that I did initially as a graduate student were simply based on using water-only fasting to improve the efficacy of cancer management. The idea being that the deactivation of the signaling cascades promotes cellular resilience, so we can protect mice against chemotoxicity. So the side effects associated with almost all the drugs that we constantly use to treat cancers. When we simply we can protect mice by simply putting them on a water-only fast, and the fast mimicking diet does the same thing. So animals are protected against chemotherapy, and what I'm showing here is essentially the management of the actual tumor progression. So you can see the top bar and the middle bar, or top graph and middle bar, are both a mouse breast cancer model. The bottom one is a melanoma model, and then the top is cyclophosphamide, and the middle graphs are on doxorubicin. Both of them are drugs that are commonly used in the treatment of breast cancer. And you can always see in both cases here, the top ones is obviously the untreated, unrestrictedly fed mice. Tumor progresses, and then you can see in comparison the treatment with cyclophosphamide or doxorubicin, which is obviously blocking tumor progression. But it's what is remarkable, I think, is when you just look at the fasting mimicking diet. In this case here, 3 cycles of the fasting mimicking diet is as efficient as treatment with a chemotherapeutic drug. So we essentially, without having a toxic insult on the body, can reduce tumor progression. The most noticeable effect is then obviously when we combine these two, and you can see really the tumor progression tapering off here. And then at the end of the study, 40 and 42 days, you can You can see this summarized here. So the advantage of the fast-mimicking diet is you can implement it in almost any treatment regimen. It's available, and it's actually independent of the cancer type. We have not seen a tumor where this didn't work, both in mouse and humanized cancers in these mice. So all the results that we had in mice ultimately led us to decide that we wanted to start a clinical trial. And the initial idea was to sort of like demonstrate safety and feasibility, but also demonstrate that it's really something that can have an impact on markers associated with health and longevity. So what you see here is we have about 100 subjects that were consented and enrolled, and then there were random. It was a randomized crossover trial. So we had 48 subjects that ended up in the control arm. And control in this case really means they could eat whatever they wanted. We did not check for that. Compared to 52 subjects that were randomized into the fast-mimicking diet arm. And then the idea was they would do 5 days of this fast-mimicking diet for 3 consecutive months. So after these 5 days, they could eat again whatever they wanted. So 5 days on, 25, 26 days, their diet, then do our diet again, and back and forth for 3 cycles. We measured, we collected blood at baseline before the diet, immediately after the first cycle, which is sort of like what are the immediate effects of this fast-mimicking diet on the metabolism, and then we did sort of like a little washout period where we collected blood and did their measurements after the 3rd cycle, 5 to 7 days of normal caloric intake. And the subjects in the control arm were matched for that, so they have the same 3-month period that you would have here. The subjects in the control arm were then ultimately allowed to also start the fast-mimicking diet. And we did another follow-up after 3 months to see how many of the effects actually persisted at this time. With regards to the baseline characteristics, you can see that, as can be expected for most clinical trials, that the percentage of female volunteers was higher, so about 60%. The average age was about 42, 43 years old in both arms. With regards to body weight, education years, and body mass index, they were comparable. I'm really only presenting the data for the 3rd cycle here, or after the 3rd cycle. Again, at this point, the subjects already have been resuming normal caloric intake for about 5 to 7 days. And at this point, we still see effects on body weight. It was an average 5-pound reduction after these 3 cycles. And again, at this point, already been eating. There was a reduction in abdominal fat, a reduction in waist circumference, a little bit over an inch. The lean body mass was preserved, which I think is important because a lot of the low-calorie diets will actually reduce lean body mass. In this case, we were able to preserve it. We see an impact on IGF-1. IGF-1, again, the component that mediates the effects of growth hormone. There was a reduction in both systolic and diastolic blood pressure, which also with a lot of low-calorie diets goes in hand. Not dramatically large changes in total cholesterol and LDL. We see a reduction in C-reactive protein, which is obviously sort of like a pro-inflammatory and associated with cardiovascular disease. And within a small population of the volunteers that we had, we actually were able to measure stem cells within the peripheral blood. And we see this transient increase here during the diet where the number of stem cells actually increases. And then after the diet, decreases again. And that's matching what we saw in animals as well. I think what is my favorite part of the study, I guess, we sort of like, after the completion of it, we looked back. And technically, we had enrolled healthy volunteers. But so when we look back at it and sort of like stratified them with regards to risk factors for metabolic disease, BMI, systolic blood pressure, and so on, we were able to categorize them and put them in different categories, just to see whether or not there were differences in the at-risk population. And you can see that I think it's pretty impressive that people that are in the higher risk groups experience greater benefits after the three fast-moving diet cycles. So you see, for example, that normal weight subjects under 25 sort of like did all right, but there was not a big difference compared to the overweight. But then really you see these changes here in the obese, right? Over 30, dramatic changes in the BMI. Systolic blood pressure over 120, higher reduction than the one in the normal range. Diastolic blood pressure over 180, higher reduction than you would expect in the normal range. IGF-1, for which there isn't really a clinical relevant cutoff, although there's multiple studies indicating that IGF-1 levels over about like 210, 220 are associated with a higher cancer risk. So in our case, we actually used the top quartile, which happens to be the subjects over 225. Again, we see a reduction in IGF-1. Glucose, the subjects over 99, so these are technically considered pre-diabetic, experienced a greater reduction in glucose after the 3 fasting diet cycles, and actually all of them returned back into a normal glucose range. So I think this aligns nicely with some of the data that we see in mice. We see a greater reduction in triglycerides, reduction in total cholesterol in subjects over 199, LDL in those people with cholesterol over 199. HDL actually no significant effects. And then CRP, again, similar to what I pointed out before, we see benefits here in the subjects that were in a range over 1, which is sort of like a moderate risk group. It returns them back into a normal range. So what I've demonstrated is that some of the findings that we have in mice are particularly, for example, reversion of type 1 diabetes, type 2 diabetes, symptoms of multiple sclerosis, and cancer management, so protection against chemotoxicity and sensitization of cancer cells to chemotherapy, are efficient. The diet is efficient there, so we're now trying to expand all these into really clinical trials, and there's currently a little over 20 trials going on worldwide that utilize this fast-moving diet. So I'll have a feeling I'll get a lot more data thrown at me. There is USC is running a trial, for example, on breast and prostate cancer. There are studies focused on multiple sclerosis running in Germany. Universities in the Netherlands do studies on breast cancer using the diet. There's trials evaluating work performance when people are on the diet to make sure it's not affecting how you work, because a lot of the people that actually do the fasting diet tend to do it during the week because it seems to have less impact on their social life, which I guess is not surprising. So in summary, and I realize I'm a little fast, but that's all I have, sorry. I showed you that nutrition, genes, longevity, and chronic disease are not separate fields, but they're all interlaced, and they're all important to be interlaced because only really together and when you put them in context, it makes sense to look at them. Diets, among other lifestyle factors, are a powerful tool to prevent or at least the onset of many of the chronic aging-related that we see, so cardiovascular disease, diabetes, cancer, and so on. I would also argue that dietary recommendations should align with healthy aging diets to maximize the long-term benefits versus short-term fads. Nowadays, a lot of the research is almost instantly available on social media and on the internet, and people tend to self-educate, and I think that's not always to a benefit. I think when making dietary recommendations, we should really consider what the long-term effects are, and if the recommendation aligns with what we would consider a healthy longevity diet. We commented briefly on the genetic variations in the centenarians, and I gave you an outlook of the experiments that were upcoming in humans with the fasting diet. disclaimer and acknowledgements. I'm supported by the American Federation for Aging Research, so thank you very much for that support. The lab is supported by the Creating Cures Foundation, Glenn Foundation, the NIA, V Foundation, Bakewell Foundation, Donner Foundation. Collaborators, the diet in the clinical trials that I mentioned was provided by Nutra. And with that, I'm done. So thank you very much for your attention.